यहाँ से अपन अब चलिए। Good morning, Dean sir. Can we start now?
Ma'am, good morning, ma'am.
Yes, sir.
Yeah, ma'am. Just wait for 4 minutes, ma'am. 11:5 we will start, ma'am. Already Sir has joined, no? Varsi Sir has joined.
Yes, sir. Yes, sir. Yes.
Sir also wants check it out, the slides, whether it is uh,
Sure, sir. Sure.
able to share it in the this one. Huh? Okay,
Okay, sir.
right.
Good morning, sir. Varsi, sir. Can you listen?
Yeah, good afternoon, ma'am.
Yeah, good afternoon, sir. Shall we start now, sir?
Yes, okay.
Oh, Dean sir, shall we start?
Yes,
Yes, ma'am. Yes, ma'am.
Sure, sir.
yes. Now, yeah. Are you able to see the my slide?
Yes, sir. Your slide is visible.
Okay.
Sir, good morning, Varshi sir.
Yeah. Good morning, sir.
Yeah, sir. Sir, I am Dr. Sheikh Abdul Rahman, sir, Dean School.
Okay. Yeah, yeah. Most
Yes.
welcome. Thank you, sir.
So I'm happy to see you, sir. And so today's is day three scientific session. Before going to deliver your presentation, madam will okay start this session and introduce about yourself, sir. Huh?
Okay,
Okay, sir.
okay. Yes.
Okay.
Good morning, everyone. I would like to request everyone to just mute your mic for a time being. Very warm welcome to all of you. Uh, Dear delegates, esteemed speakers, sir, faculty members, research scholars, participants, and everyone joining the third day of our 5-day International Faculty Development Program on Drug Design, Delivery, and Development. I'm Dr. Shikita Mishra, Assistant Professor at School of Pharmacy, Galgotias University, and it is my pleasure to be your moderator for your today's session. Before we begin, on behalf of our organizing committee, I extend my heartful gratitude to all the participants for their enthusiastic involvement over the past 2 days. We are very happy and it's all our pleasure to host you all. We hope you find the discussion insightful and enriching. So, for today, it gives me immense pleasure to introduce today's eminent speaker, Dr. Musrat Hussain Warsi. Associate Professor in Department of Pharmaceutics, College of Pharmacy, TAF University, Kingdom of Saudi Arabia. Dr. Musrat has over 10 years of teaching experience and 1.5 years of industrial experience. He holds a PhD in pharmaceutics and industrial pharmacy, has made outstanding contribution to the field of novel drug delivery, computational modeling, pharmaceutical technology, pharmacokinetics, and bioequivalence studies. He has authored over 90 publications, contributed 14 book chapters, and presented more than 50 papers at international and national conferences. Dr. Musrat also has 4 patents and has handled multiple research products funded by TAF University and DRDO, and serves as guest editor for MDPI General Pharmaceutics and editor-in-chief of Biological Sciences. We are truly honored to have him with us today to share his deep expertise and research insights. Without taking much time, let us now begin today's session. Please join me in welcoming Dr. Musrat Hussain Warsi for his enlightening talk. Sir, the virtual stage is all yours.
Okay, thank you for your introduction. So, a warm welcome to all of you for this faculty development program, and very good morning to all of you. So, today I will deliver a talk related to the nanomedicine enabled drug delivery, and it mainly focus what is the opportunity related to the nanomedicine challenges what we face, and most important, what is the different risk assessment related to the nanomedicine. So first we will start with the general definition of the nanomaterial and the nanomedicine. So as per the International Standard Organization, we define the nanomaterial when we are going to change the dimension of any existing particle into the nanoscale range. It may be external dimension or internal structure surface dimension. So generally we talk, when we talk about the nanoscale, so we talk about this scale should be in between 1 to 100 nanometer. So when we talk in the different delivery system, it may be 150 or 200 nanometer, but exact property of any material which is going to change, it should be less than 100 nanometer. So as per the European Commission and the International Standard Organization, we have to go for the different assessment when we are going to reduce the size in the nanoscale. And when we are going to apply the nanotechnology in the field of medical, then we say this is the nanomedicine. So what the nanomedicine and how nanotechnology is going to adhere with this nanomedicine. So generally we say that nanotechnology, which refer how we are going to control and manipulate the any material in a nanoscale dimension. So this is not a simple combination. This is a combination of science, engineering, and technology to achieve the scale at a nano size range. So when we monitor, repair, and control these nanoparticulate material for the biological system, then we refer this as a nanomedicine, and this is going to engineer for the nano device or structure or nanoparticles. So this may include like the nanomaterial for the biological devices, or nanoelectronic biosensor, or molecular nanotechnology, all which are going to affect the biological system that refer to the nanomedicine. So why we say nanotechnology, nanomedicine, nanoparticle, nanoemulsion? Why this nano? Why this nano scale is very special for us, and what the different opportunity we can achieve by this nanoscale dimension? So we can say this is the scale where a lot of biology occurs. What does this means? It means that like when we talk about the different molecular component of our body, like the DNA strand, this in the nano dimension means this is around 2 nanometer size is diameter. Hemoglobin, this is around 5 nanometer diameter. The pores in biomembrane by which different molecule is going to process transport. So there are the different pores like the microporous, the size is less than 2 nanometer. Mesoporous, the size is between 2 nanometer to 50 nanometer. and microporous, the size is more than 15 nanometer. So we can see here the different biological macromolecule component are just deal with the nanoscale range. So if we are going to modify our external particle material engineered in the nanoscale, so definitely it will alter the biological system. So what the different opportunity we can get from here, like enhanced permeation. So by manipulating the size in the nano dimension, we can achieve the higher permeation rate. So when we talk about the permeation, as we know for the bioavailability, because our ultimate goal for any drug delivery is bioavailability, to enhance the bioavailability. And how can we enhance the bioavailability? We know that the BCS classification system, that is the biopharmaceutical classification system, is deal with only two things. What is that? Solubility and permeability. So, like the class one, both have the high solubility and high permeability. But when we move the second, third, and fourth class, either it has the low solubility or high permeability, or low permeability, high solubility, or like the fourth, low solubility, low permeability. So all is going to affect the bioavailability if it is not has the high solubility and the high permeability. So by the nanoscale, we will achieve the large surface area. So we will achieve the high solubility as well as permeability. So by this we can enhance the permeation through the blood circulation and also by the different biological membrane. So by this we will achieve the higher bioavailability. Same like the enhanced interaction. At the nanoscale, the particle, there the exposed surface area, there the more chemical reactivity and biological reactivity, and more interaction will happen inside the biological system. And enhance retention. Also, it will help to retain these nanoparticulate system inside the cells because it is going to escape from the natural body defense mechanism and it can be enhanced. So we can achieve all these parameters which is important to achieve the higher bioavailability. And also we can achieve the passive targeting like the EPR effect, well known, and mainly deal with the tumor cells. So that is the enhanced permeation. So this particle will come out from the leaky vasculature system and it will be effectively retained inside the tumor cell and show their efficacy. And also we can control the release and we can modify the release also by which we can achieve the higher ability. So we can see what is the different opportunity we can get by this nanoscale dimension. Next is this is the scale where the quantum effect rule on the behavior and property of the particle. So what is this quantum effect? And this is more related to the physics, quantum physics, and this is just what we say that one particle can exist in the form of wave also. So when we are going to reduce the size in the nanoscale and when we talk about less than 50 nanometer, so it is going to show some type of quantum effect and we cannot say we certain with this property. So at a nanoscale, any particle can show the effect on different effect on the fluorescence, conductivity, melting point, magnetic property, chemical activity. So we have different figure as compared to the normal microparticles. As we can see the example of gold nanoparticle. Gold. Everyone aware about this. And gold, what is the color? That is the yellow color or pale yellow color we can say. But when we are going to develop the nanoparticle and when it come in the nanoscale range, this color is going to change from yellow to red or purple color. And this property is also used for the diagnosis of the tumor. So when it going to convert into the nanoscale dimension, its particle is more prone to movement and in presence of light it show the different activity and it start to show the color change from yellow to purple color. And also due to the PR effect, it is going to accumulate into the tumor cell and easy to monitor and diagnose the tumor, especially related to the pancreatic tumor and other type of tumor. So we can say this in the 30 nanometer or nanoscale dimension, we can achieve this diagnosis with the help of gold nanoparticle. So we can see this by modify the property, by modifying the drug release, we can achieve the diagnosis, means imaging of the different organ, as well as we can achieve the enhanced drug delivery, modified drug delivery in the controlled fashion. So we can see all these opportunity we can achieve with the help of only nanoscale dimension. So this is the scale which mainly deal with the surface and interface. Because we can see here when we are going to reduce the size, as here in this picture we can see when the surface size is 1 centimeter in the cube, so one dimension is 1 centimeter, so total surface area is 6 centimeter square, but the cube dimension is going to reduce to 1 millimeter cube, then we achieve this surface area 60 centimeter square, means 10 times more. But further, when we are going to decrease this diameter of cube from 1 millimeter to 1 nanometer, what the characteristic we can see here, the surface area. We can see there are 60 million centimeter cube. Surface area is going to increase, means 1 million times increase in the surface area. So we can assume what is going to happen. By increasing the surface area, it is more exposed to the outer environment, and there will be more chemical reactivity, and more it is more surface will be exposed, there will be more solubility. We can modify the surface by like the pegylation or certain antibiotic reactivity, that we can go for the targeted delivery, and we can functionalize this surface because more surface area is exposed. So we can see by reducing the size in the nanoscale how much surface area is going to expose, and this surface area is more important determination for the activity of any particles. So we can say this nanoscale is very important, and this give a lot of opportunity. So here we can conclude that by nanomedicine, what can we offer? We can increase the solubility of the drug for poor soluble drug. We can fostering the transcytosis, means we can enhance the transcytosis process, which can engulf the drug particle from outer surface to in and deliver to the inner surface of the membrane. We can control the release of the drug, and by targeting particular organs and protecting it from the different body system, we can go for the target drug delivery and reduce the toxicity related to the drug. Also, we can increase the pharmacokinetic parameter of the drug by increasing the enhanced bioavailability, and there will be the increased Tmax, concentration, Cmax, and other, and we can achieve the PR effect. We can easily deliver the combination of drug. Also, we can protect the drug against the body defense mechanism, because in the normal delivery, there is the loss of drug by the different body defense mechanism. Also, by visualizing the location, we can take the image and we can diagnose the problem, and also we can identify the malignant cell via the molecular detection. So we can see there is a lot of opportunity by what we achieve by the nanomedicine. So here there is the certain engineered nanoparticle, and their application is given. So we can see here like the silver material function biocides, it's used in the wound treatment, odor control, prosthesis, titanium dioxide, the well-known material, and this is used for the cosmetics, pharmaceutical, sunscreen, iron oxide, that is the super paramagnetic. Also, it is used in the biomedical and electronics, quantum dots, carbon nanotube, and dendrimers. So these are the different materials, and we have already prepared and well developed stabilized nanoparticle, and they have the different application in the drug delivery, healthcare issues, and other type of problem. But we have a lot of opportunities, so why we need to worry about these engineered nanomaterial risk? So the main factor is that we have a lot of research related to the development of nanoparticle or nanoparticulate based drug delivery system. But we have a very lesser research and little information related to the hazard and what is the dose response and what is the exposure of these nanomaterial to the human and the environmental factors. Also, these nanoscale affect all the different property like the conductivities of the particle, melting point, magnetic permeability, chemical reactivity. So we have to little information about these. What is the different behavior of each parameter of the particle? Also, we are going to develop this, so this will change in the nanoscale, the property is going to change. And when we reduce the size less than 10 nanometer, in that case, it is start to show the quantum effect. And that effect is uncertain. We don't know it is going to show the effect of particle or it is going to show the effect of wave. And also maybe there's sometime dramatic changes in the release of the drug and their exposure. For example, sometime nanoparticle it is going to disperse in water or biological media, it is going to agglomerate. So overall nano dimension is going to lose and overall release will be disturbed. So there is the chances of some risk related to these nanomaterial, and these risks we will go further about this. Now, what is the nanomaterial properties? So when we talk about this material surface area volume ratio, when it is going to increase as the particle is going to small. So when the surface area volume ratio is going to increase, the interaction of these particle with the surrounding atom, surrounding tissue is going to increase. And more reactivity will be there. So overall behavior of the particle as well as surrounding tissue will be changed. And we already know that when particle is going to more means a lesser size like the less than 10 nanometer, it going to start to show the quantum effect, and quantum effect is totally uncertain. So here we can see that there is a certain example how the characteristics of any particle is going to change when we are going to change its dimension from micro scale to nano scale. For example, copper. Copper is opaque. This is not transparent. But when we are going to develop the copper nanoparticle in the nano scale, it is going to change from opaque to transparent. Aluminum. This is very stable material, but in the nanoscale range, it is going to become combustible. It is flammable. Platinum, that is inert. But in the nanoscale dimension, it acts as a catalyst. Silicon, that is the insulator, well-known insulator. But in the nanoscale, it is well conductor for the electricity. Gold, this is solid, but in the nanoscale, it turns into the liquid. So we can say how the property of any material is going to change at the nanoscale. So these nanoscale and nanomaterial risk assessment, we can have a lot of challenges, and how first we have to know the basic things, how we have to define these nanomaterial, how we have to measure the exposure. So first, how do we define the nanomaterial? So generally, we define the nanomaterial when this nano dimension is less than 100 nanometer. So at this, its property is going to change as we discuss about the copper, aluminum, like that, gold. And sometime we have to differentiate it between single particle, or this is agglomerate or aggregate. So single particle means they are the different nano particles. But when we talk about the agglomerate and aggregate, like we develop the formulation. So simply this is agglomerate. When we disperse, it will all the particle will be dispersed because it is just joined or combined together in the loose by the van der Waals force. So by dispersing in the water or suitable media, it will disperse easily. But when these nanomaterials are aggregate, because they are going to rigid bind or very tightly bind to together, so it is not going to disperse easily in the biological medium. So it will show the different property as compared to the single R agglomerates. So we have to define accordingly. This is the single particle agglomerate or aggregate. And also what the transformation is going to happen when we are going to change the chemistry. Next is how do we measure the exposure? So nanoparticle or nanomaterial, we have to measure what should be the unit. This should be mass or this should be number or surface area. So this depend either we have to measure in the mass or number or surface area. This depend what type of nanomaterial is there and what is more prone to expose. So simply we measure in terms of surface area because in the nanoscale dimension, surface area will be more exposed. There will be more surface area as compared to the number and the mass. And also we have to quantify, we have to measure what is the lower limit of detection for that nanomaterials. Also we have to differentiate the engineered materials and their background. Sometimes their background is going to change, so the combustion at the nano size particles. And the most importantly, there is no suitable data, or still we have a lack of data related to what is the fate, what is the transport and uptake mechanism, and these are going to strictly adhere with the impact of the solubility, what is the coating and surface charge, all these about this nanomaterial and how it is going to transport and how it is going to show the toxic effect, because sometimes it is going to accumulate, or in the lower nanoscale it is going to show another effect what we are going to predict. So these nanomedicine challenges, because they just have a lot of opportunities, so they have provided the benefit. So apart from benefit, also they have certain risk. So in the nanomedicine, there the increased efficacy as well as unpredictable risk is there. So we have to identify after delivering these nanomaterials into the human body where it is going to locate, what is the exact geography of biological response, and this is very important in case of nanomedicine. So we have to know how we are going to deal with this. So here we can see the cycle for the nanomaterial properties and toxicities. So here first is the shape. So when we talk about the shape, the toxic interaction with the cell, and it can be enhanced due to the shape of engineered nanomaterial. So the shape is also very important when we are going to engineer the nanomaterial, like the sphere shape, it is good as compared to the fiber one. Aspect ratio, like fiber tend to be more toxic as compared to the particles size. So smaller, as we are going to lower in the nanoscale range, it will become more toxic as compared to the larger particle. Surface area, as the reactivity of engineered nanoparticle is more when the size will be less, because if size will be less, nano dimension will be less, there will be more surface area, it will be more exposed to the outer environment, and there will be the more reactivity. Next is the chemical composition and the crystalline structure. So these toxicity depend upon what is the chemistry of engineered nanomaterial, and it is the crystalline form or what is the other form of the crystalline or amorphous. And the surface coating. So by surface coating, it may change the physicochemical property of engineered nanomaterial, and it may affect the toxicity issue also. So these are the different characteristics which govern the toxicity of engineered nanomaterials. So in this picture, we can see what is the different toxicity mechanism of nanomaterial. For example, the component or the drug which are incorporated inside the nanomaterial are the different constituent by which we have prepared nanomaterial, how they are going to release any other contaminants from the environment. They are going to adhere as a vector, and they are going to release with the nanomaterial constituent release. Physical effect of the physical effect of the size and shape of the nanomaterial, and it will show the inflammatory response. Also the surface reactivity, because surface is exposed, so in the presence of light it will more reactive, and all these combined going to affect either the biological membrane, either the gene expression, either the macromolecule, either the enzymatic activity inside the body. So we can say all these factor is going to affect the different features of our body. So all these are going to enhance the toxicity. So we have to analyze all these factors critically. So here we can see the oxidative stress model, and this is the most popularly model for the oxidative stress. So here we can see when we are going to deliver the nanomaterial to any patient, any persons, living organism, there is the generation of ROS, reactive oxygen species. So we can see in the normal stage, there will be the higher ratio of glutathione, reduced glutathione to oxidized glutathione. So there will be no oxidative stress. It is the normal. But in the tier 1, which we can see in the orange color, when the reduced glutathione is going to little bit less as compared to the oxidized glutathione, so there is the antioxidant defense mechanism will start inside the body, and they will cope up with the oxidative stress. But further more reactive oxygen species will be generated, then it will move from tier 1 to tier 2. And in this case, there is the excessive generation of MAP kinase and NFKB cascade, and there will be the inflammation in the body. And further, more oxidative status will be there. There is the lower ratio of reduced glutathione to the oxidized glutathione, and it will come in tier 3, and there will be cytotoxicity called the apoptosis and the cell death. So we can see how it is going to affect the oxidative stress for the cell death. When there is the excessive oxidative stress due to the generation of ROS system, due to the nanomaterial consumption. Here another example is given for the pulmonary toxicity pathway. So when nanoparticle is going to expose, it lead to the oxidative stress, and for the pulmonary system, when there is the ROS system, it is going for the downstream signaling. It will activate the cytokinin growth factor cascade, and it will act as the receptor of tyrosine kinase. Then further generation of TNF alpha, interleukin, TGF beta 1, and called the fibrosis. So there the cell will be fibrous. And another way, it is directly going to the DNA, damage the DNA, and cause the mutagenicity in the nucleus. So we can see how these stress, which is going to generated by the nanoparticle exposure, affect the cell and cell organelles. So here is the different test which I have summarized here to assess the toxicity of nanomaterial. So there are some in vitro studies, and after confirming with the in vitro studies, we have to go for the in vivo test. So in vitro test, most probably the genotoxicity, oxidative stress, inflammatory responses, increased cell death, cell death, alteration, and penetration of barrier. So followed by that, more confirmation we have to move for the in vivo test, that is the increased deposition by the inhalation method, altered clearance, inflammation, oxidative stress, formation of fibrosis and granulomas, translocation to the other organ, and precancerous lesion. So all these tests will be essential to determine the toxicity behavior of nanomaterial. So we have to go before commercialize our formulation or before going for any conclusion, before for the scale up, that all these tests meet either this is the positive side of the toxicity or the negative side of the toxicity. So we have to assess what is the right outcome of these tests. So here the summary of human effect of nanoparticle and nanofibers are given. So when we are going to give the nanoparticles, either it will go to the brain, either it will go to the lungs and cause the inflammation, and further there is the asthma, lung cancer, or chronic obstructive pulmonary disease. If it will go to the blood, there is it will either it will go to liver or spleen. Either it will cause the thrombogenesis, atheromatous plaque, or autonomic nervous system. It cause the cardiovascular death. So these are the fate. We can say it maybe happens. What exactly it is going to happen, we don't know. So we have to analyze every factor associated with these, and we have to characterize what is the risk factor. Same like with the nanofiber, it will directly go to the pleura, inflammation, and cause the chronic obstructive pulmonary disease or asthma. So here is the approach is given to address the nano risk assessment challenge. First is the nanomaterial risk assessment framework, which screens the life cycle analysis, comprehensive environmental assessment, what is the expert judgment, bridging toxicology, high throughput screen toxicology, and what is the predictive toxicology. So we will go one by one here. So here is the health impact framework for the engineered nanoparticle and what is the source. So when we talk about the source, so when we are going to develop the engineered nanoparticle, so what the material we are going to use, how it is going to expose with the environment, and what the different other contaminants of environment is going to adhere with this nanomaterial, then how we are going to deliver this by inhalation or by the injection or by the oral route or by the dermal. So it is going to change the different properties. So in human body, either by the brain, bone marrow, heart, kidney, liver, lungs, all through, it will go and impact the health system. Either it will go to affect the airway inflammation or allergy, the cancer, cardiovascular disease, cytotoxicity, developmental toxicity, mutagenicity, immunogenicity, or toxic organ toxicity, thrombogenesis. So any type of health impact may happen. So we have to analyze every time before develop to the engineer nanoparticle with the raw material after developing the nanomaterials, and we have to go for every factor very critical manner. So in this nanoparticle risk assessment in the framework 1, so we can see there is a cycle how first we will identify the hazard system, and that hazard system is what chemical composition, what the composition we are going to take. Then what is the particle size? What means final particle size of our formulation? What is the structure? I mean, this is the spherical shape or the fiber shape, and what is the coating? If you are going to coat, what the coating material, how much coating we are going to achieve. Then we have to go for the exposure assessment and the toxicity assessment. In exposure assessment, we have to check the particle behavior, what is the use, for which purpose we are going to develop the engineered nanomaterial, and what is the durability and stability of nanomaterial, and which receptor system it is going to interact and bind, and what is the route of administration. We are going to give by the inhalation or through the oral route, parenteral route, or topical route. And for the toxicity assessment, we have to check all the pharmacokinetic data. What is the absorption, distribution, metabolism, excretion, and we have to check reactivity and the dosimetry. Then we have to characterize the risk. What is the effect? Means apart from the therapeutic effect, what is the another effect? And what is the nature and extent of these effect? And how can we control this effectiveness? So all this parameter we have to check for the risk assessment of nanomaterial. In the framework 2, we have to go the individual characteristics of the nanomaterial, like particle characteristics, concentration matrix, chemical composition, exposure route, translocation mechanism. And we can see by this framework every characteristics, like the particle, like the surface area, particle shape, it is going to agglomerate, or what is the particle size distribution, concentration in terms of number, mass, or surface area, chemical composition, or the route of administration, all is going to interlink together. And they are going to affect the different parameter like the what is the pollutant emission, environmental concentration, exposure to the human, biological relevant dose, all are interlinked together, and they are going to affect by affecting the oxidative stress, and these stress either they are going to affect the cellular level or the gene level, and finally if they are going to affect the gene level and they are going to affect the health, and sometimes it will cause the cellular death also. So we can see the cycle and these framework by which we can assess, and this is very complicated, not a we cannot say very easy task. This is very complicated and we have to study it is over a periods. So on this assessment we have to go for the expert judgment, and we have to categorize this, what is the exposure and what is the toxicity area. So when we have to expose this product, the nanomaterial, means how we are going to use and how we are going to dispose, and based upon different particle characteristics we have to go what is the exposure, that is the presence of nanomaterials into the body, and what is the uptake property of the body for these nanomaterials which come for the exposure, and there is the transport and toxic effect. So this come in the toxicity also. Transport and fate means how it is going to transport, because we know there are the mainly three type of transport like the endocytosis, transcellular, paracellular. So what the mechanism is going to pick up by these nanomaterial, and how it is going to dispose inside the body organ, and how it is going to secrete, how it is going to accumulate, we have to assess all. So these transport and fate come under both criteria, exposure and toxicity. And after that we have to measure ecological, means what is the risk for the ecological system as well as what is the risk for the human system. So this toxicity effect module, we can see here all the characteristics of the nanomaterial, like the chemical composition, surface reactivity due to the surface area exposure, surface coating, adsorption tendency, all this going to affect the changes at the cellular level or the subcellular level. And when there the change in the cell level or the subcellular level, it is going to affect the different parameters inside the body. Like sometimes we says for the irritation effect or immunological effect, neurological, hepatotoxicity, genotoxicity, allergy, or fetal effect. If there is the irritation, this irritation may damage to eye, or lungs, or gastrointestinal tract. So all are related to the human health risk. So what is these main parameters? So here we can see chemical composition. What is the different constituent we have to take to develop the nanomaterial, what is the active materials, what is the size, because this is the nanoscale, so size every time matter. As we are going to decrease the size, it moves more toward the quantum effect. So this is when it moved towards the quantum effect, it means this the property of nanomaterial is unpredictable. We cannot say this is the property. It may change. And also the more surface area, there will be the more surface reactivity and more absorption. So apart from giving the opportunity like the more retention, more permeability, change the release behavior, these are the opportunity. But apart from opportunity, these are the risks associated with nanomaterials, which we have to also consider every time when we are going to develop any novel nano formulation. So based upon this, we have to apply the expert judgment by adhering with the nano risk assessment. So what is the risk trigger? What is the use? For which purpose we are going to develop the nanomaterial? How we are going to dispose this nanomaterial? And other most important things, the nano product information. Nano product information means what we have discussed earlier about the each and every characteristics. So we say like pre-formulation means what we do before the formulation, like we measure every aspect of each ingredient that is compatible or not, like what is the pH, partition coefficient, solubility. And after that, when we develop, we go for the size analysis, morphology, surface exposure. All these information we have to measure very critically, and then we have to go for getting the information, what the information is missing and what the information is needed to stabilize the risk ratio as compared to the benefit ratio. And on the basis of that, we have to relatively risk rank. We have to rank the risk. This is the this risk ratio is more as compared to the benefit ratio. Our risk ratio is less as compared to the benefit ratio. We have to rank it on the basis of expert elicitation. So here one predictive toxicological approach is given. One example I have provided here, and this example is reference to the titanium dioxide nanoparticle. So I think everyone is aware about this titanium dioxide nanoparticle, and this example is included here because this is the most commonly used nanoparticle in every field, not only the drug delivery. It is used in the cosmetics, sunscreen, toothpaste, heart, optics, electronics, photocatalytics, anti-ultraviolet light agent, food packaging, water treatment facilities, medication delivery system. So it is used multi-purpose use of this titanium dioxide nanoparticle, and this is very stable, anti-corrosive, and has photocatalytic properties. So you can see this titanium and the structure of titanium dioxide nanoparticles, and this size is around 50 nanometers. So this titanium dioxide nanoparticle is used to study over the Cyanobacteria elegans, that is a roundworm, to respond to the acute exposure to sublethal concentration. So here we use the concentration of titanium dioxide, that is 20 parts per million. That is the sublethal concentration for the roundworm, and we expose this for the 24 hours. And for analyzing the toxicity of this titanium dioxide, different behavior of roundworm was studied. And what was that properties, the characteristics of roundworm, like what is the speed of movement, what is the reversal phenomena, omega band, and turn count. So all these had been studied before the exposure and after the exposure. So you can see this is the picture of this roundworm. So now what was the outcome of the studies? So when scientists studied the behavior of these roundworm nematodes, they found the behavior is totally altered when exposed for the 24 hour with the titanium dioxide nanoparticle. And it was found that overall suppressive behavior was obtained for the roundworm. And this suppressive behavior is due to the induced neurotoxicity. So they observed, they found that the neurotoxicity, this is the due to the titanium dioxide nanoparticle. And due to this neurotoxicity, the behavior of that roundworm is going to change, it is going to decrease. So these findings include the toxicological information of titanium dioxide nanoparticle, and this is very useful to manage manage and to determine the risk of potential hazard to the organism as well as ecosystem. So what we can conclude by this study, that what can be ethically assumed and accepted with the risk potentially posed to the human subject with the novel therapy or new treatment, it should be reasonable in relation to the therapy and potential benefit along with the regulatory standpoint with respect to the risk which is subject to the society, and it should be minimized whatever whenever it will possible. So we have to consider all the things, because we know there is the chances of risk, but this risk ratio should be lower as compared to the potential benefit. And when we have to develop these nanomaterials when there is extremely needed, then we have to develop and we have to deliver by stabilizing all the toxicity related information. So finally, the concluding remarks for this topic, that nanomedicine and nanotoxicology are two sides of same coin. So we cannot complete the coin only by developing the nanomedicine unless until we are not going for the other aspect of the coin, that is the nanotoxicology. So we have to develop safer and more biocompatible nanomaterial through the material design and engineering. So what the nanomaterial we are going to develop, it should be biocompatible and safe. So what is main objective? It's safe rather than effectiveness. First, we have to main emphasize on the safety and then go for the effectiveness. And these nanomedicine during the developing, we have to integrate with the concept of safety by design. So there is a lot of machine learning tools are available. So we have to integrate these not only for the formulation development parameter, also the toxicity related issues with the quality by design method. And also it is important to characterize the nanomaterials for the different prospects about the particle size, their roughness, the shape, charge, composition, surface coating, whatever the parameter possible, each and every parameter we have to characterize. And further, we have to develop and implement the toxicity testing protocol, and that accurately predict the potential toxicity of nanomaterial in the living organism. And finally, the therapeutic use of nanomedicine require a different framework which balances the therapeutic benefit against the potential risk of the harm. So this is the most important criteria. So what the medicine we are going to develop, it should be stabilized in both benefit assessment along with the risk assessment. And also we have to implement the effective safety measure during the every process step, either this is during the production process or handling or the disposal of these engineered nanomaterial, and we have to implement this safety protocol not for only for the human as well as for the environmental aspects. And also we have to increase the public awareness and understanding of these potential risks which is associated with the nanomaterial, and going to implement the regulation and guidelines for their safe use. Also, we have to increase the research into the toxicology and environmental impact of these nanomaterial to better understand for these effect and development of effective remediation strategy. So apart from only develop the drug delivery system, we have to also move toward their toxicological aspect as well. So, in summary, we can say that reducing the toxicity of nanomaterial, it requires a combination of improved material design, better toxicity testing, increased safety measure, increased public awareness, and what is the ongoing research is going. We have to focus all these aspects, and we have to stabilize in terms of regulatory standpoint, quality standpoint, and other different international law framework. So this is all about my presentation. So we can say when there is the unorganized, non-collaborative, non-cooperative research, so we can say we cannot say we are going to do in the right direction or not, and we cannot achieve a lot. We can achieve a little bit, but we cannot conclude anything. So any research it should be organized, collaborative means we have to collaborate with every type of professionals, either formulation scientist, toxicologist, pharmacologist, and other related scientists. And we have to, by collaborating, we have to work in a cooperative manner. So when we will do, we can achieve a cake. So yeah, this is all about my presentation. So thank you to all.
Thank you so much, sir, for such an insightful and inspiring session. Your explanation and research perspective have truly broadened our understanding to the innovation of drug design and development. Everybody talks about making the nanoparticle formulations and doing everything, but nobody talks about the toxicity and the
Yeah, thank you.
requirement required for the nanoparticles. That was very insightful and very knowledgeable session.
Thank you, ma'am.
Now we will open the floor for the short question answer round.
Okay, sure.
Participants are requested to post their question on the chat box or raise their hand for interaction.
Hello, sir. This is Dr. Sabba. Can you hear me?
Yes,
Yes,
ma'am.
ma'am,
Yeah.
you are audible.
Yeah, sir. The session is awesome. Excellent. Excellent session.
Okay.
So far, I'm
Thank
into
you.
the nano drug delivery system. Actually, I have prepared nanoparticles, nanofibers, etc. But the toxicity point of view is so good. So, for this, do you have any reference for this fluorescent nanoparticles and toxicity, sir? Later toxicity?
Actually, fluorescent nanoparticle also show the actually these fluorescents are related to the light scattering and all this. So these are we have to go through the ICS guidelines. There is the I don't remember exactly what is that para for the phototoxicity issue. So we have to go for that ICS guidelines. I
Okay,
think
stability
that
guidelines, Q1 R3.
yeah, I think yeah,
Mm.
QN3. And apart from that, when we have to go for the in vitro and in vivo study, as I mentioned in the presentation, so when we have to deliver these in the rat or mice, what you are going to develop the formulation, so we can go there. The live imaging machines are available now, so we can capture this how much it is going to accumulate in particular organ like the liver or kidney or anywhere. So by this fluorescence, we can find out because this is fluorescence based, so we can easily find out. Because if it
Okay,
is going to accumulate, it will show the more toxicity, and we have to determine how much time it is going to accumulate. Because of course you are going to develop the nano formulation, so it will stay in our body for a long time, but it should not be stay for a very long time that it can be dangerous for any organ.
Yeah, the another thing, another question for me is, like you say, we have different nano drug delivery system like nanoparticles, nanofibers, nanorods, etc. So among all the drug delivery system, which one will have some more toxicity, sir? Which drug delivery system do you have come across?
Nanofibers. Actually, nanofibers, the shape is long, elongated type. So it shows the more thrombus and more toxicity as compared to the nanoparticle or nanoemulsion. Because still a lot of research remain related to stabilize the toxicity about this. But as per the literatures, it's so more toxicity as compared to the other.
But still, people are using nanofiber. They say
Yes.
electrospun nanofiber is good.
Yes,
Even we
it's
have
using
manufactured
because
electrospun
this is
nanofiber.
the academic research. As we
Uh,
know, this is the academic research, and academic research we can show anything.
because they can
Yeah.
also be used for a cardiovascular disease. Stent material can also be prepared using this nanofiber. So, how
Yeah,
far
but
you
that is the
say the
depot type.
toxicity,
There the
even
difference.
when I heard your lecture, the nanoparticle is good than fiber. The toxicity wise, you have shown the priority, order of priority.
Again,
Ju-
again, ma'am.
no, sir, the priority order, just like that you have, in your lecture, you have just spelled out. Like nanoparticle is good than the nanofiber material, as well as nanorods.
Yes,
So the toxicity wise, the nanofiber is higher.
yes,
So, but still people are using for cardiovascular disease. So that's what I just want to hear from you.
yes, ma'am. Actually, for the cardiovascular disease, they are using because they are using as a depot and targeted one because they are not going to circulating all over the body. First thing is that, and I have discussed here. We have to analyze the risk benefit ratio. So if
Mm.
it is going to more benefit as compared to risk, because when we talk about the any disease like the cardiovascular you tell, so sometime we have to deliver the drug or in the exact location for a different time point, and if you are not going to achieve that, so risk is more. So what the risk related to the fiber we have to calculate, and then we have to deliver. So I think in that case we can deliver, but as per my opinion, it should be avoided.
Oh, yeah. Thank you, sir. Thank you. Thank you so
Okay, you're
much.
welcome.
Thank you.
Okay,
I'll be in touch with you, sir,
you're
for
welcome.
further collaboration work.
Yeah. Okay,
Okay,
sure,
sir.
sure. You're
Yeah,
welcome.
sir. Thank you. Thank you.
Good morning, sir.
Yeah. Good
Dr.
morning.
Dr. Mamta Bishnoi this side. It was very nice session, very informative, sir. So
Thank you.
my question is, what type of toxicity evaluation we should go for when we are preparing any type of nano formulations? How we decide what type of toxicity we should evaluate? Like in case of cytotoxicity, immunotoxicity, and all these. So when we go for characterization, what type of toxicity evaluation we should add?
Yes, ma'am. First, it depend upon which type of formulation you are going to develop. Means for which disease you are going to target, and you for which route you are going to give. This is oral route, or by parenteral, or by the topical route. So accordingly, we have to go for the different toxicity test. Like suppose, for example, if you are going to give by the topical route, so you have to first check the toxicity in the skin. So
Right.
we have the different histopathological test, and we have to go for the histopathological analysis, and then for what the organ you are going to targeting. So for that, you have to go and analyze that organ for the healthy animals, and then you have to analyze in the treated one, and the different kits are available based upon the chemical reagent. You have to analyze what is the different chemical component it is going to change, and what the ratio should be changed as compared to the standard one. Then we can stabilize information related to these toxicological aspects.
Okay, sir. Thank you so much, sir. Thank you.
Hello, sir. So, am I audible?
Yes, yes.
Yes, so myself, Jagdish. So, I have a doubt regarding where you talked about this quantum effects of this nanoscale, and where in the slide you're showing about the gold nanoparticles. They were not yellow, but they appear in the color of red and purple. So,
Yes.
we know that, like, normally we are working in nanoparticles. So, the surface plasmon resonance plays a very important role for this color shift from the yellow to like purple brown or whatever. So whether the color change is directly proportional to the size of the nanoparticles, like on the first stage itself, whether we could be able to identify whether the size of the nanoparticles formed would be like aggregates or it has been agglomerated, or does it depend upon the type of nano materials which are preparing? For example, if I'm going to use for silver nanoparticles, whether the color change or the shift is directly proportional to the aggregates, or is it just corresponds to gold nanoparticles? So, in simple, I could just tell that whether when we observe the color shifts, we could just determine the shape of the nanoparticle. I mean, the size of the nanoparticles. Is it possible, sir?
Yes, sir. Actually, what I understand from your question, because sometimes your voice was disturbing. So, like when we talk about the size, when we are going to decrease the size, and we can say less than 50 nanometer, less than that. So, thus color is going to change from pale yellow to the purple or red color. So, when we talk about the agglomerate or aggregate, so agglomerate is okay because when we develop and when we go for the centrifugation, it will agglomerate in the bottom. But if it is going to aggregate, it is very tough to redisperse in your either into the your medium or biological fluid or whatever you are going to disperse. So it will not going to change the color. But if it is going to redisperse, so it is going to change the color. So you will find out that easily that color is going to change. It is agglomerate, not in the aggregate form, because this color is due to the size. And if we are not going to get or achieve that particular size, the color will not show, because this color is based upon the reactivity of size in the presence of light. So they are going to change the color in the presence of light, and this color changes depend upon the size. So if it is agglomerate, then when we will redisperse, we will get that purple color. But if it is not going to redisperse properly, we will not get the uniform red or purple color.
Okay, sir. Okay, sir. Thank you. I get it.
Sir, there was one more question in the chat box that
Okay.
when we take drugs, they might get broken down into nanoparticles. So that could be the reason of toxicity of the drugs.
Again, ma'am, when take the drug or when take the
When
nano?
we take, no, when we take the drug, it is written in the chat box that when we take the drugs, they might get broken down into nanoparticles. So that could be the reason for the toxicity of some drugs.
No, actually, when we take the drug, means suppose what I understanding, if you are
Yes,
taking the
sir.
tablet, it will go and break, but it will not
Yes,
break in the nanoparticle. Either it will solubilize, and when any drug is going to solubilize, it will be in the solution form. There will be no particle,
yes, sir.
so it will not break in the nano form. If it is going to break, then there is no need to develop the nano formulation.
I think Rukmini ma'am must have got her answer.
Okay.
Okay, sir. Sir,
Ma'am, good morning, ma'am. Actually,
good
you've
morning.
been reading that question for me. It's very thankful for me. Just I
Right.
want to prolong my question.
Hmm.
Is it the man, sir, has been discussing with all the toxicities, right? And
Hmm.
even he has been focusing on brain and other perfused organs for the body, but he didn't tell me the risk factors related to topical buns, the dermal wear, so like nano emulsions or nano emulsions which had been applied onto the skin.
Yes, ma'am, of course, you're welcome. Yeah, this is not only for the different organ, every part means when we are going, but when we compare from the internal body part, this is less when we
Yes,
compare,
sir.
but
Yes,
definitely
sir.
there will be toxicity.
Yes,
And
sir.
so why we go for the toxicity test for the dermal also.
Yes, sir. So can
Yeah.
we have a, I mean, is it in a large scale the toxicity occurs, or it is in a primary stage that we can use for the little bit use? I mean, maybe so 2 to 3 years for psoriasis or any other diseases related to topical acne, like any other urticarias or for different types of diseases to the topical ones. So we can just use it for the small, shorter period rather than the longer period, right?
Yes, and but when we are going to develop for the longer period, so we have to go for the chronic toxicity as well, because
Yes,
we are
sir.
go for the acute, yeah, acute
So majorly,
toxicity.
what are the of toxicities we can expect for this topical dermal systems?
We have to first see what is the change in the dermal structure,
Yes, sir.
epidermis, endodermis, and like that. And then we have to analyze the different layers, how they are going to behave. And
Yes,
by this, it will give the idea if it is going to rupture, are there the change in the cell structures of
yes, sir.
the, then it will give
So any
a lot of
particular test
idea. Then we
that
will
we
go in
can,
the first.
so
Yes.
any particular test that we can test for this type of toxicities available in the market?
This not particular, yeah, because first we have to go for the simple, the living organism. We take the
Yes,
skin
sir.
and go for the test. This is the very
Yes,
normal test.
yes,
Yeah.
sir.
Then we have to go for the oxidative stress test.
Okay,
So that oxidative stress also tell a story for this chemical damage or not. So if there is a
okay, but
reduced
the skin
glutathione,
sensitive test goes for that, isn't it?
yeah, sorry.
Skin sensitive test.
Yes, yes.
Yes, sir. Thank you. So thank you for
Okay,
your information, sir. It has been very good for me.
okay, you're welcome.
Thank you very much, sir, for
Okay.
patiently addressing all the questions. For
You're welcome.
the due to the less time, I'm now blocking. There are so many questions in chat box till now. Then
Okay.
I hope everybody can contact to you directly. And now uh, I would like to thank you so much for. I would like to now present to you the certificate of appreciation on behalf of our Department of Pharmacy, Galgotia University. I take this opportunity to express our heartfelt gratitude and present this certificate of appreciation as a small token of our respect and thanks for your valuable contributions.
Okay. Thank you. Thank you, ma'am.
We are truly honored to have you with us today, sir. Thank you once again.
Okay, you're welcome, ma'am.
And
Thank you.
all the participants, for you, there is a glimpse of what is going to be for tomorrow. Before we conclude, I am sharing the link of feedback form to the chat box as well as to the YouTube chat box. Everybody can do their feedback, give their feedback. And for the glimpse of tomorrow, as tomorrow we will continue our series of expert talk focusing on emerging technologies in pharmaceutical sciences with more engaging sessions and hands-on insight from the distinguished speaker. As Professor S.K. Abdul Rahman sir will talk about development of nano herbal formulation and its evaluation. And Professor Gaurav sir will enlighten us about the tools, softwares, and database for the structure-based drug design. So we encourage all the participants to join us on the time and continue this journey of learning and collaboration. I thank everybody. Um, so now, sir, you can,
Should I
as
leave,
you
ma'am?
have, yes, sir, yes, sir.
Okay. Thank you, ma'am.
Thank you so much, sir.
Okay, welcome. Thank you.
On behalf of all our Gulgotia Parivar, we are
Oh,
very thankful for you.
okay, you're welcome, ma'am. Thank you.
Thank you, sir.
Ma'am, can we leave?
Yes, ma'am. Everybody who has got the feedback form can leave. I was
Yeah, I
just
filled up the feedback
making it
form.
continue
Thank
so that everybody can get the link of feedback form.
Okay, ma'am. Thank you so much.
Ha, sure. Thank you. वो आने देते